Still many unanswered questions that I was telling you about. And while we were doing all of this work, there's sort of a revolution in what we know about P. acnes. So here's little P. acnes with just the star. And, some great work that was done by Julie Segre and Elizabeth Grace and others looks at the microbiome of acne. And if you look in these sebaceous prone areas, I think many of you know that P. acnes kind of rules the roost. It's the most prevalent in the sebaceous areas of the skin. Work from Jenny Kim's lab shows that within acne lesions, you have increased expression of toll-like receptor 2 on inflammatory cells. You also have increased expression of the NLRP3 inflammasome, so you have increased inflammasome activity as well. Work from Fitzgibbons at UCLA identified that within the P. acnes, there are many different ribotypes. So ribotypes, there's a lot of different ribotypes of P. acnes. And they looked at people that have acne over here, 48 of them, and people that don't have acne, 51 of them. And just looking at the colors, you can see there's a lot of pink and orange here. And these are the ribotypes 4 and 5 that seem to be predominant in the folks with acne and less predominant in the folks without acne. So this raises the question as to whether the ribotypes 4 and 5 are causative for acne, or maybe they're just better suited to live in an acne environment. So we're not really sure. And there's a lot of interesting studies being done on that. And that leads to the question of, what is P. acnes doing in the follicle? It obviously rules the roost there. It lives there. It's its home. Is it there to fight off other bacteria, other pathogens? Is it trying to win the war against staph, or what's going on there? Another interesting work from Rich Gallo's lab and from other labs looks at the metabolome of bacteria on the skin. So the bacteria are very metabolically active. They're making all kinds of small molecules. But what's the relationship between what the bacteria are doing and how does that affect what the host cells are doing? So is there some crosstalk, some communication? So there's a lot of interest in the microbiome world in expanding into metabolomics and trying to figure out what the relationships are between small molecules made by bacteria and the host. And if you can think of some analogies there are certain types of bacteria that if you were to measure them in the skin or in other places, they're always together. And I can't remember the names. They were bacteria I never heard of. But they sort of have a symbiotic relationship. Like one bacteria can't make a certain type of amino acid, but the other bacteria can. So or metabolite. So they live with each other, and they are able to, its survival is promoted by its presence with the other one. So what's going on in acne? Is there some sort of relationship between the bacteria, the host cell, the immune system, maybe even fungi and viruses that might contribute to the whole process? So I think it's a fascinating area that we'll hopefully look in. So this you might remember from a commercial, I forget, some insurance company. So is P. acnes the defender of the follicle? Is it really defending us from other pathogens, or is it really causing a problem? And these are some questions that I think are fascinating and hopefully research will answer as we move forward. So this gets to the topic of the meta-omics of acne. Like, who are the players? We know that the host, not everyone gets acne. Everyone has P. acnes. So why is that? Is it that there's differences in the immune response to the P. acnes? Is it the P. acnes itself? Are there certain ribotypes that are more causative? So very interesting. And now that we have whole genome sequencing, we can do these types of studies, looking at the presence of fungi and viruses as well. And how do all of this interact with your environment, with diet, hygiene, and exposures? There's a fascinating study, I think it was also done at UCLA, that looked at, I think it was vitamin B12 metabolism. Did you see that one? They found that if you were to, they looked at vitamin B12 metabolism in P. acnes bacteria, and they then they gave patients vitamin B12, and they found that when patients were supplemented with vitamin B12, and they looked at the P. acnes, the P. acnes downregulated its production of B12, which I thought was pretty fascinating. So is there some sort of metabolomic type interaction between even diet and the bacteria on your skin? So a lot of unanswered questions. So I've talked about trying to figure out how drugs work, and the traditional bench to bedside that I'll touch on very briefly at the end sort of looks at molecules on cell systems that go into animals, that go into people. But there's a whole new way of drug discovery that uses computational biology. Many of you might be familiar with that. Many of you may have heard of a concept called drug repurposing. So now that we have a lot of gene expression data, we have it in human skin cells, we have it in cells in culture, so there are a lot of data sets available of different types of skin diseases, and a lot of data sets for drugs. Drug companies, when they're doing large-scale screening, they will use all of their compounds in a library and put it in cell systems and generate data sets of how it changes gene expression. So if you can take the data sets from cell cultures with libraries of drugs and take the data sets from patient disease and data sets from patients on drug for a disease, there are ways to build a network model of a disease. So we're collaborating with folks from Sinai and support with the Acne Cure Alliance to work on developing a network model for acne. So our goal is to generate data sets for understanding the acne disease process and identifying novel therapeutic targets by drug repurposing. So as part of this, there's two parts of the study. The folks at Sinai are working on comparing normal patients to acne patients. So we're doing facial microbiome using whole genome sequencing and packed biosequencing. We're doing biopsies of the back using RNA-seq as a more comprehensive method of looking for gene array expression. We're also analyzing blood. So this will be done for acne folks and people without acne. Our part of that is we are doing sort of a state-of-the-art version of the study we did back in 2008. So we're looking at how isotretinoin generates effects on a variety of things, and we're going to take that data set, merge that with the acne model, and then have that data set be available to companies and anyone that's looking at compounds and seeing if there's some match in terms of effect of a compound on the cell system that might play into the model for acne. So our portion of the work is we are looking at the effects of isotretinoin on the microbiome, gene expression in the skin and the blood. We're also looking at DNA methylation of genes because with the long-term remission with isotretinoin, you have to wonder if there could be some gene silencing that's going on. Is something getting turned off that is important in acne? We're also looking at gene expression in keratinocytes, pseudocytes, and fibroblasts, so generating a lot of data sets. So we have 3 different cohorts of patients that are enrolled in this study. Everyone gets a biopsy, a blood assay, and a microbiome assay at baseline. One cohort of 6 people had all of this done again at 1 week. Another cohort had it done in 4 weeks, 8 weeks, and then everybody gets it at 20 weeks, and everybody gets sampled at 6 months. So we've completed everything, and our last cohort, we're just collecting the 6-month specimens on our last 6 of the 18 people. So we're hoping to have that data set pretty soon. So that's sort of our kind of computational model that we're looking at to try to find other compounds that might be helpful for the treatment of severe acne. And my last quick case is more of the traditional bench-to-bedside. So we know that sebum production is a problem in acne. Isotretinoin and oral contraceptives and maybe some laser-type treatments are the only things that really make a difference for sebum production. So an example of sort of the bench-to-bedside that our laboratory works on, and we've done this for many different compounds for many different companies, and I'm just going to use one as an example. So to produce sebum, you need to make a lot of fat, a lot of lipids. And I'll give you this example of acetyl-CoA carboxylase is an enzyme that converts acetyl-CoA into malonyl-CoA, which is the beginning of the synthesis of fatty acids. So there's a drug you might have heard about. It was published in the JID and also in, I think, the JAMA. This drug inhibits this enzyme, acetyl-CoA carboxylase. So this drug was studied in an animal model for sebaceous glands, looking at the sebaceous glands in the ear of a mouse, and also studied in human pseudocytes. That we did this in our laboratory, and it was also done in another laboratory using primary pseudocytes. So for our assay, we have our pseudocytes in culture, and we want to see what kind of lipids are being made that are analogous to sebum. So all lipids are made from 2 carbon fragments of acetate. And so if we have a 2 carbon fragment and one of the carbons is labeled with C14, we can just put all of the C14 labeled acetate fragments on top of our cells, and our cells have all the enzymes that are needed to make all kinds of lipids that you see in sebum. So that's basically what we do. We give C14 acetate, put them on cells, incubate them. The cells have all these enzymes, and then we measure how much C14 label comes out in these lipids that are present in sebum. And then we add the test drug to the system to see what lipids basically get blocked or shut down. So we did this with that with the acetyl-CoA carboxylase inhibitor, and basically we found that in our in vitro system that it did reduce the production of sebaceous lipids, and that's going to come out in the JID. What you might have seen in, I guess it was the JAD, was this drug went from the testing into phase 2 clinical trials, a small trial done in 53 patients on drug and 55 in vehicle, a phase 2A, and there was data to suggest that it might possibly be beneficial in the treatment of acne. So I think it's going into phase 3 studies now. So that's just an example of the traditional sort of bench-to-bedside type of translational work. So basically, I just hope to share with you today a little bit of what we do in translational research. There's lots of different forms of translational research. Hoping to learn more about isotretinoin so we can find better drugs, and trying to identify new drugs that may suppress sebum and improve our patients with acne. And I think that's it. Support I've had from the Dermatology Foundation, from Penn State, from NIAMS, other agencies as well. Thank you.